Hello and welcome to our lecture dedicated to apoptosis and necrosis, which are both forms of cell death. Before looking at cell death in detail, let's recapitulate what we have learned in our session dedicated to the cell fate. As you can see here, and hopefully as you remember after watching the videos within the last session, our cells can basically survive, die, divide, or differentiate, and all of those processes are quite tightly controlled. Cells require signals continually from the surroundings to survive, so our cells are only surviving if they are sensing certain signals from the outside. However, they are always ready to respond to death signals, and today we are going to be looking at cell death in detail. So our cells can basically survive or divide. If they divide, they need to grow first. And what you need to keep in mind here, that cell growth and proliferation are actually not the same. Cell growth involves an increase in cell size without an increase in cell number. Cell growth involves synthesis of new proteins and membrane lipids. Also, during this phase, there is an increase in the number of organelles, because obviously during division the resulting cells need to have all of the organelles required for a cell to function properly. Cell growth also may involve replication of the DNA, but this is not always necessary. Similar to other cell fates, cell growth is controlled by certain signals from outside of the cells, and here specifically growth factors are important. Growth factors control cell growth positively. In addition, contact with other cells regulates cell growth as well, but in a negative manner. So all of our somatic cells are negatively regulated in respect to the cell growth by neighboring cells. And this mechanism of control is often lost in tumors, where there is an uncontrolled cell growth. After the cell has grown, usually proliferation or cell division follows. This always involves replication of the DNA and division to produce two resulting daughter cells. It usually follows from a cell growth phase, and if not a reductive division will occur, the proliferation always results in one or more smaller cells, because obviously cytoplasm needs to divide as well. Another cell fate we discussed in our last session is differentiation, and differentiation is a quite specific cell fate which only stem cells can do. So this process includes specialization of uncommitted stem cells into terminally differentiated cells that can fulfill the function within complex organ system. This usually, but not always, involves them ceasing to proliferate. The last cell fate type we had a look at is apoptosis. Apoptosis is one of the forms of cell death we are going to be looking at today. So, what you need to keep in mind here is that cells are always ready to respond to so-called death signals. In order to initiate apoptosis, no new gene activity and protein synthesis is required. And lastly, this is a highly controlled and organized process, unlike other types of cell death we are going to be looking at, for example, necrosis. So how do our cells decide what they are going to be doing? Regulation of cell fate required a combination of very, very different signals. So different combination of signals that a cell is sensing will lead to different cell fates. And also the output is dependent on the cell type. For example, certain combination of growth factors and other signals will produce one cell fate in a neuron, but another cell fate in, for example, a liver cell. Again, the same signal combination can lead to cell death in one cell type, but for example, proliferation or differentiation in another. And this is something you need to keep in mind. Lastly, I showed you that deregulation of basic cell fate regulation mechanisms often contributes to development of cancer. Here, cancer often develops if cells become self-sufficient in growth signals, so they can survive and grow even though there are no external growth signals present. Also, in cancer cells, we often observe an insensitivity to anti-growth signals. Cells within cancer often evade apoptosis, and they also do have limitless replicative potential. They are potentially immortal, and this is something you need to keep in mind. So, what are the learning outcomes for today's lecture? This lecture is gonna be fairly straightforward, and after watching all of the 3 videos within the session, you should be able to describe and contrast the features of apoptosis and necrosis. You need to note the steps and signaling events controlling apoptosis, and lastly, understand the events that result in necrosis, which is another type of cell death. So, why is cell death important at all? First of all, cell death, and not only proliferation, plays an important role in embryogenesis and fetal development. Cell death is also important if a cell is damaged beyond repair. Cell death plays an important role in removing cells infected with viruses, and this is something we are going to be looking at in our lecture focused on immunology. This controlled cell death can prevent viral spread. It is also important in removal of ineffective or potentially damaging cells within the immune system. But again, this is something we are going to be looking at in detail in our immunology lecture series. Lastly, cell death is important for the control of malignant cells, for control of cells that underwent tumorigenic transformation. Those cells can often be recognized by the immune system, followed by initiation of cell death. As we've heard already a few times, there are 2 types of cell death, and the first type of cell death is necrosis. Necrosis is fairly uncontrolled cell death and is always associated with pathological processes such as disease. In contrast, apoptosis is programmed cell death or cell suicide. Apoptosis is an essential part of normal health and also development. In average, adult between 50 and 70 billion cells die through this process per day. So if you do your math, then you'll realize that in a year, your entire body weight of cells will have died through apoptosis, and those cells are usually replaced. So what is the result of necrosis versus apoptosis? What you can see here on those slides are electron microscopy pictures of cells that undergo necrotic and apoptotic cell death. Obviously, they are quite different cell death types, even if we look at simple morphology. In apoptotic cells, we see formation of tiny vesicles, and we call them apoptotic bodies. In contrast, in necrotic cells, we don't see those vesicles, and the content of the cell is released into the body and usually induces inflammation. So in apoptosis, those vesicles produced are taken up by macrophages, and as a result, there is no inflammation in the system. And this is something quite important, as we will see in the following sessions as well. I hope that you remember that I already showed you this very video, and this shows cells in the dish, and some of those cells will undergo apoptosis because in this very experiment, signals that induce apoptosis have been added into the medium. So if you look at those cells, then you will see that there is obvious change in morphology of the cells. If you look at this cell, then you will see that there is formation of those apoptotic bodies. This cell started with the apoptosis. Those cells were slightly later, but the result is always the same. You see quite controlled cell death resulting in formation of apoptotic bodies. In this video, you can see apoptosis once again, but in a slightly larger magnification. But again, here it's quite fairly easy to see that the result of this type of cell death is formation of vesicles that can be then taken up by macrophages. So, why is apoptosis or controlled cell death important? As I mentioned before, apoptosis plays a crucial role in development of an embryo. In this slide, you can see the formation of a hand. So in A, those cells between the fingers are still present, but during development are getting removed via induction of apoptotic cell death. And indeed, this not only happens in animals, but also in humans. During development, the fingers are developed fairly late. In first instance, those cells between the fingers are still present and they need to be removed via apoptosis. Similarly, apoptosis plays an important role in brain development. Nerve cells rely on establishment of connections. So all cells that are not able to establish correct connections with neighboring cells during development are getting removed via apoptosis. Therefore, only correctly wired cells survive and contribute to brain development. In addition to embryonic development, apoptosis is also fairly important for the immune system, because it destroys cells that are self-reacting. This prevents autoimmune disorders. But again, we will have a dedicated lecture on immunology later on. So apoptosis is also important in the immune system because it contributes to removal of cells infected by the virus. And here, quite defined death signals play a role. But again, we will hear details of this process in our lectures dedicated on the immune system. Lastly, apoptosis is important to keep the system, the physiological system in a homeostatic state. So apoptosis is important for homeostasis. It is a counterbalance to cell division, and it's also important for removal of old and damaged cells. So this is all what you need to remember in terms of importance of apoptosis. Lastly, apoptosis is also a drug target, but this is something you will hear a lot more about in part 3 and 4. Many drugs that actually are used for cancer treatment make cancer cells susceptible to induction of apoptosis again. But again, this is just additional information. So, how is apoptosis triggered? There are two main pathways for triggering apoptosis. The first way to induce apoptosis is the so-called extrinsic pathway or receptor-mediated pathway. Here, so-called death signals bind to receptors, followed by activation of downstream signaling cascades. The second pathway leading to activation of apoptosis is the so-called intrinsic pathway. And this is usually activated by cellular stress. For example, experienced by cells during chemotherapy, can also be induced by ultraviolet light, and so on. So what are stages of apoptosis independent of the way of activation? So this process is basically the same for the extrinsic and intrinsic apoptosis. If a healthy cell senses death signal, both extrinsic and intrinsic, This leads to a commitment to die, and this is still reversible. This is something you need to remember. However, if this stage is surpassed, execution of apoptosis starts, and this step is irreversible. Our dying cell now condenses, and the proteins within the cell become cross-linked. Later on, the apoptotic bodies form that you have seen in the videos, and they are now ready to be engulfed by macrophages and also by neighboring cell. Because all of the components of the cell and of the cytoplasm are now embedded in those apoptotic bodies or apoptotic vesicles, this way of cell death usually does not lead to inflammation. With this, I'm ending the session number 1, and I will see you soon in our next video.